memory clinic. So this is a 66-year-old female who presented to the memory clinic reporting a decline in her short-term memory, executive function. She also had word-finding difficulties, impaired balance, and frequent falls. And these symptoms developed in 2015 and have progressed since then. Past medical history was significant for a diagnosis of a stage 3B adenocarcinoma of the lung in May of 2014. She received systemic chemotherapy for this and radiation treatment. And in 2015, she had an MRI of her brain after having some worsening dizziness that revealed some right and left parietal lobe and right frontal lobe masses consistent with metastatic disease from the lung cancer. She received whole brain radiation in December of 2015, and then a boost to the residual lesion in 2016. And then she was started on palliative nivolumab, which is one of these new checkpoint inhibitors in January 2017. And miraculously, since then, has had no interval change in her metastatic disease in her brain. So at her initial evaluation, we did a MoCA, and it was 25 out of 30. She lost some points for the clock, fluency, and delayed recall. She just had a high school degree, so we gave her an extra point for education. And she had a DUCE score of 42 out of 50. She had very minimal functional impairment. It was rated at 6 out of 36 on our functional activities questionnaire. And her main limitations were in driving. She basically stopped driving. No family history of neurodegenerative disease, and her neuro exam was normal, except for some decreased sensation, light touch in her right lower extremities, an impaired postural response, and impaired semi-tandem stance. I diagnosed her with mild cognitive impairment, likely secondary to the whole brain radiation treatment, but couldn't exclude the possibility of another underlying neurodegenerative disorder. I recommended that she start an exercise program, Mediterranean style diet, and we also started a trial of memantine. So I saw her a year later for follow-up. She still reported some progressive changes in her short-term memory, but she felt that with the memantine, her memory, her thinking was clearer. And she had repeat MRI by her oncologist that showed no progression of her CNS metastatic disease, and her repeat MoCA was unchanged at 25 out of 30. So memantine and CNS radiotherapy. So we know that there is progressive cognitive impairment, particularly in processing speed and executive function in patients who receive a whole brain radiation therapy. It typically follows a biphasic pattern with a transient decline early on in the treatment, followed by some improvement and then progressive impairment that occurs months to years later. It's thought to be due to vascular insufficiency and infarction with accelerated atherosclerosis and microangiopathy related to the whole brain radiation therapy. On her MRIs, though, there was no evidence or no significant white matter disease that was noted, which you might see in the setting of radiation-associated CNS treatment. There was a 2021 systematic review that looked at 4 prospective clinical trials of memantine during radiotherapy, and they found that there was better performance on the Hopkins verbal learning test, delayed recognition at 6 months, and longer time to cognitive decline with memantine treatment. And so at Duke, radiation oncology has actually started using memantine prophylactically in patients with reasonably good expected survival. And they've also modified their whole brain radiation approach to try to avoid the hippocampus, to avoid some of these long-term cognitive sequelae of radiation therapy. And so to conclude, as patients live longer with cancer, especially with the development of these targeted therapies and checkpoint inhibitors and other immunotherapies that are able to allow patients to have stable disease for longer, it's important that we think more about the neurocognitive sequelae of cancer treatment, particularly when it comes to whole brain radiation. And so focal radiation or whole brain radiation with hippocampal avoidance are approaches that can decrease the side effects associated with this therapy. And memantine treatment during and after radiation treatment may also decrease neurocognitive adverse effects. And there are actually several clinical trials that are ongoing to evaluate the efficacy of memantine during and after CNS radiation. And so just have my references here. And with that, I'll turn it over to Andy.
Thank you, Daniel. You're one person too quick. That's a very interesting case. I had no idea. So to introduce Andy today, our fearless memory disorders division director, Dr. Kim Johnson, will do the intro.
Thanks, Rich. So just want to introduce Andy today, who's a very valued member of the memory division. Andy grew up in Lawrence, Kansas, and a favorite memory of his growing up was waking up at 4 a.m. to go fishing with his father, which must be why he's always the first person in clinic every morning. He attended the University of Kansas for undergrad and met his wife there, where they both majored in biochemistry. He was a basketball fan at Kansas, which served him very well this past year when he won the Memory Division March Madness competition by picking the ultimate winner of the tournament. Then after undergrad, he attended Chicago Medical School at Rosalind Franklin University and then did his residency in neurology. So he's been part of the memory division since August of 2018 after he finished a 2-year behavioral neurology fellowship at UCSF. He's now an assistant professor of neurology and has a dual appointment in the Department of Pathology. He's been very active academically with multiple publications, about 10 in the past 2 years since he's been at Duke, which is just phenomenal. He routinely lectures to neurology residents and founded and leads the Neurobehavioral Conference for the department. He's also a valued mentor and always has a medical student, resident, or fellow in the clinic with him. So I would say that in addition to all these academic accomplishments, Andy's also a very caring and compassionate provider and definitely a team player in the memory division. He routinely receives glowing comments from patients and was designated as a neurology all-star in March of 2021. So Andy has so many varied research interests. He's very curious, interested in aging, inflammation, epigenetics, cognitive effects of COVID, AD biomarkers, and retinal imaging. And he also has a special interest in the relationship between tuberous sclerosis and frontotemporal dementia. So I would say that one thing Andy always tells me is that he's lucky to be at Duke, but I think we're very fortunate to have Andy here in the memory division. So today he's going to speak to us about his interest in tuberous sclerosis. So take it away, Andy.
All right. Thank you, Kim. Is everyone able to see my screen now?
Yep.
Okay, great. So thanks for the introduction, Kim. The title of my talk is really linking neurodevelopmental disorders to neurodegenerative disorders. As Kim mentioned, it was tuberous sclerosis complex. And what I'd like to start off with is just my disclosures. I have very few, so this is it. And this is just the outline of my talk. The majority of it's going to be just really kind of setting up the entire current study here at the bottom. So I'm going to introduce TSC and FTD and the overlap between the two and kind of where I'm going from there. But before I start, it's early in the academic year, so I'd like to always just kind of define what dementia is, so we're all on the same page. People who have seen my lectures before have seen this table, but I think it's always useful to review. This is how I define dementia. It's a clinical term that really says that the patient or the caregiver is saying something is wrong cognitively, behaviorally, or psychiatrically, and it's impairing their ability to live independently. So that is what dementia means. It does not tell us what the etiology is, but really just affecting the ADLs and IADLs is really key. And then, of course, they're going to be testing abnormally on cognitive testing, whether it's the SLUMS, the MMSE, or the MoCA. It will always be abnormal. Now, I'd like to go on to the neurodevelopmental disorder, tuberous sclerosis complex. So I want to introduce it first. It was actually described several centuries ago in 1835 by a French dermatologist who really described the angiofibromas, so these outpouchings around the nose. And that was one of the hallmark features of TSC and remains so today. It was recognized finally as a genetic disorder in about 1910. And then the mutations in TSC1 and TSC2 were finally discovered in the 1990s. So TSC1 is known as hamartin, TSC2 is known as tuberin. These mutations actually lead to the dysfunction of something called the mechanistic target of rapamycin, mTOR, the mTOR metabolic pathway. And I just want to give you a quick overview of the mTOR pathway. It's a very complicated figure, and the TSC 1 and 2 are boxed in red. But what it comes down to is that TSC 1 and 2 act as the brakes in cellular proliferation. So you take the brakes away, the cells just continue to divide out of control, which is why there's so many masses that are involved in TSC. Epidemiologically, TSC, the incidence is about 1 to 6 to 1 to 10,000. The prevalence is about 1 to 20,000. So not the most common disorder that we've ever seen, but people are living longer, and that's why it's being more recognized. There's actually 2 ways to diagnose TSC. The first one is a genetic diagnosis. So a patient must have a pathogenic mutation in TSC 1 or 2 independent of the clinical findings. So in the clinic, there are some times where you don't even have to see the patient. As long as they have a known pathogenic mutation in these genes, you've already made the diagnosis. So pathogenic mutation is really defined as a mutation that alters the protein product of TSC 1 or 2. And so this includes nonsense, frame shift mutations, large deletions, or missense mutations. But just be warned that 10 to 25% of TSC patients have no identified mutation. So you can't hang your hat only on the genetic testing, because a normal result does not completely exclude TSC. That's why there's a clinical diagnostic criteria as well. They're really made up of major and minor features. And this combination of major and minor features really can fulfill the clinical criteria. So it's 2 major and 1 major, 2 minor. I won't go into it, but there's a mixture of both that fulfills that clinical criteria. And as I had mentioned before, TSC really involves several organ systems. Here on the left, this is the CNS involvement. So subependymal giant cells. This is something that shows up in the right exam or your board exams for neurology, subependymal nodules and retinal hamartomas. And then you can see on the right, this is a list of major features in various organ systems. The heart, lymphangioliomyomatosis, otherwise known as LAM, is actually a lung mass. And then renal angiolipomas, and then a slew of kind of dermatologic findings as well. So the next slide is an interesting one. This is a registry here at actually done in Europe. And what they did was just categorize when certain masses show up at different ages. So you can see here is color coded up here. And then boxed is just the CNS involved type masses. And what I thought was interesting is that the SEGAs typically are the most worrisome for neurologists, and they don't come up until probably the around 10 or preteen ages. The SCN, subependymal nodules, and cortical tubers, they're kind of throughout early stages of life as well. And the retinal hamartomas are similar to SEGAs, where there's kind of a peak really at the age between 9 and 14. And I just want to show you a few radiographic features of the CNS involvement in TSC. So right here on the left, you see a subependymal giant cell astrocytoma, a SEGA, right at the foramen of Monroe. It really, this is very dangerous because it can cause acute hydrocephalus, and that is an emergency. Another one here is the subependymal nodules, as shown by the arrow right here. It's just, this could just be a continuum of a SEGA. It's just at the very early stages. The next one is the retinal astrocytic hamartoma. So it's shown in this white circle here. And then in panel B, it's the same mass, just using fluorescein angiography to kind of better characterize it. You can see it more kind of a 3D rendering in the B panel. Another major clinical feature is the cortical dysplasias that I previously mentioned as well. The arrows really point at the hyperintense signal on this flare sequence in the coronal plane. You can see right near the vertex that hyperintense signal bilaterally. But then the arrowhead is something I want to point out. You see this kind of white tail that goes subcortically. That's called a radial band. That is almost pathognomonic only for TSC. And then there are the minor features, which pretty much involves the dermatologic organ system or renal cysts as well. And so those are harder to diagnose because there's a lot of people who don't have TSC who also have these features too. But what's most relevant to me as a behavioral neurologist is something called tuberous sclerosis complex associated neuropsychiatric disorders, so otherwise known as TAND. This was described by DeFries et al. In 2015 and really shows three different categories: cognitive, behavioral, and psychiatric, which really defines behavioral neurology. And as you can see, there's a list of all these symptoms in all three categories. I'd like you to keep this in mind because 90% of people with TSC suffer from TAND, but only 20% are actually treated or acknowledged. It's something that's really in the infant stages in regards to what TAND is. And because I'm going to transition now to the other end of the life course, which is a neurodegenerative disorder called frontotemporal dementia. I mentioned frontotemporal dementia several months ago in my case presentation too, and so I'm going to quickly go over this as well. FTD is just a clinical term that describes three subtypes: the behavioral variant, frontotemporal dementia, BVFTD, and then the two language variants, the non-fluent agrammatic and the semantic variant, primary progressive aphasias. 60% of the cases of FTD are the behavioral variant, BVFTD, whereas the two other language types collectively make up the rest of the 40%. And I want to touch base on FTD, the terminology itself. You might have heard Pick's disease too. Pick's is just an antiquated term used to describe one of the subtypes of FTD. Specifically, it was the semantic variant as well. Epidemiologically, the average age of onset is actually quite young. So someone who's 58 or around there, maybe early 50s, late 40s, or early 60s, I'm always thinking of FTD in the clinic. Men and women are equally affected by this disorder, and the incidence of this is about 3.5 cases per 100,000. To give reference, Alzheimer's disease is about 4.2 per 100,000, so it's actually quite a common disorder. Unfortunately, it's under-recognized, and many people go to a geriatric psychiatrist or a psychiatrist first for the behavioral changes before they're referred to a behavioral neurologist. And then genetically, there's actually three common genes that are associated with the genetic form of FTD. So progranulin, GRN, C9orf72, C9 open reading frame 72, and the MAPT gene, which is the protein product of tau. I want to touch base very quickly on the neuropathology of frontotemporal dementia and point out, again, really emphasize when we talk about the neuropathology, we change the terminology to FTLD, so frontotemporal lobar degeneration. There's really three major categories. So transactive response DNA binding protein, it's a mouthful, so we just call it TDP43. There are three different subtypes, A, B, and C, depending on the morphologic features. And then there's FTLD tau, as you can see, shown by the arrows here. The red arrows are actually showing balloon neurons filled with tau. And then there's a last one called FUS. The common theme between FTLD is that there's an accumulation of some sort of pathologic protein, and that's actually a common theme in neurodegenerative diseases in general. Now, the clinical criteria to describe and diagnose BVFTD is actually, there's actually 6 of them. And so you need to have 3 out of the 6 to have a possible diagnosis of BVFTD, and they must be persistent. So someone who's had a little too much to drink and is acting a little disinhibited for one night doesn't qualify for BVFTD. It has to be over several weeks to months, even years. But that's the hallmark of BVFTD, is the disinhibited behaviors. The second one is an early apathy or lack of inertia. So these are the people that just lay around and sit around, watch TV all day when they used to be very active outdoors. There's an early loss of sympathy or empathy. So for example, if a loved one was hospitalized with COVID-19 and intubated, they would just kind of brush it off thinking, oh, they'll recover. It'd be an abnormal response. There's early perseverative, stereotyped, or compulsive ritualistic behavior. So this kind of varies widely. So I've had patients who just go check themselves in the mirror about 20 times in an hour, or they'll just take their watch on and off for no reason. These are the stereotyped behaviors that you will see with BVFTD. And then there's hyperorality and dietary changes. So there's been a lot of patients who really restricted their diets. So I've had a patient who only ate shrimp, and that's all he ate. A lot more patients will gravitate towards a sugar-heavy or carb-heavy diet. That's one of the hallmark dietary changes in BVFTD. And then lastly, this is actually a very important point, is on neuropsych testing, there's a lot of executive function difficulties with relative sparing of the memory and visual spatial function. And this is actually tells you that it's the anterior part of the brain that's more affected, the frontal and temporal lobes, versus the parietal and memory centers of the hippocampi. So that's why it's a very telling and significant feature on neuropsych testing. Now, going to the imaging part of BVFTD, I want to emphasize that you can't make the diagnosis based on imaging alone. So I can't just review someone's image and say, oh, this is BVFTD. I would probably say it's highly suspicious for FTD, but I can't definitively say it because you need to have the clinical features, is the point. Now, as you can see here, in this brain, you might have, I've shown this picture before, but the right anterior temporal region is actually, and lateral and medial are actually quite atrophic compared to the left side. This was the initial visit. 3 years later, continued atrophy of the right temporal region, and now it's actually swinging over to the left side and causing atrophy there. I also want to point out that there's orbital frontal atrophy here, which explains a lot of his behavioral symptoms, and I'll explain that in a little bit. In addition to MRIs, you can use an FDG PET, but I would use it with caution because there's a lot of confounding factors in interpreting an FDG PET for the diagnosis of FTD. Because if someone's had a history of TBIs or head traumas, that can falsely decrease the metabolism of the frontal and temporal lobes, leading you to a diagnosis of FTD. And then I also lastly want to point out the changes in the 3-year time span in these two images is actually very common in FTD. So if you start on the right side, after about 2 to 3 years, the left side becomes evolved and vice versa. So this is important to kind of tell patients what to expect. So the right temporal lobe, they'll have a lot of decreased empathy, changes in eating habits. And then when it swings over to the left side, you start telling patients that there are going to be language problems that continue to deteriorate over time. And I think that's helpful in the clinic because they can prepare for that. They know what to expect because it's unnerving when you start seeing new symptoms in a neurodegenerative disorder. But then what is the link between TSC, which is a neurodevelopmental neurogenetic disorder that happens very early in life, versus a neurodegenerative disorder, FTD, that happens much later in life. So I'd like to talk about the phenotypic overlap between these two groups. So I'd like to go back to the tanned table that I had shown earlier, the cognitive, behavioral, and psychiatric categories. Highlighted in red is the overlap between TAND and FTD. So the FTD patients show a lot of the symptoms shown in red, and you can see that there's significant overlap primarily in the cognitive domain, but behaviorally and psychiatrically as well. And this all started at UCSF when a gentleman in his 6th decade of life presented with behavioral changes. He was a very successful person who owned his own construction company, but in his 6th decade of life, he started having several sexual harassment lawsuits brought against him. And so he was brought to UCSF, and the MRI brain imaging was, as I had shown earlier, so right temporal lobe atrophy, and then progressive atrophy involving the anterior temporal, and then actually orbital frontal cortex, which explains his behavioral changes. What was also interesting is that he had a strong family history of behavioral changes late in life. He had several brothers, several other siblings who showed kind of disinhibited behavior later on. So genetic testing was done for the 3 common ones, progranulin, C9, and MAPT. They all returned negative. But when we looked at the FLAIR sequence, you saw this, as the arrow is pointing out, this kind of hyperintense signal going into the subcortical region, and that was the radial band. So TSC1 and 2 testing was actually initiated, and the novel TSC1 mutation was found. And so right here in the star, you see that the arginine was switched to a cysteine, and which led to a stop codon. So premature stop of the TSC1 protein, which led to this disorder. So this begs the question, is TSC1 and 2 a novel or genetic form of FTD? And if it does, that implicates the mTOR pathway that leads to neurodegenerative diseases. And so we started recruiting adult patients who were able to live independently to UCSF during my fellowship from 2017 to 2019. We published our findings here in this paper down below, and we excluded anyone who had another genetic disorder. It's surprising what you would find other genetic disorders that along with TSC as well. But I think the whole point is that TSC patients have a broad phenotype. They are not always devastated by seizures and fully dependent on loved ones. They can live very independent lives, and they're living way beyond the age of 50, and now they're having cognitive problems, and we don't know why. Now, for this study, the demographics included 18 TSC patients. The age range was from 27 to 66. 72% were female, and genetic testing was finalized in 10 out of the 18 with an equal number of TSC1 and TSC2 mutations. This is actually important because traditionally it was thought that TSC2 mutations showed a more severe phenotype than TSC1, which definitely wasn't the case for us. Now, the next one is the clinical evaluation. We had 3 cohorts, one with normal cognition, another one, of course, with TSC, and then the last one with FTD. The clinical testing included the TAN checklist severity score, and then the Neuropsych testing. The TAN checklist severity score really was just a whole bunch of questions that were yes, no for symptoms, and the total number of yeses were added up and then tallied and then compared between the 3 cohorts. So the average TAN checklist severity score for TSC is shown in this left column here, FTD here in the middle, and then for people with normal cognition, you can see here. So in all 3 domains, you can see that there's a significant actually difference when you compare the TSC and FTD cohorts to the normal cognition, but actually they're actually quite similar between TSC and FTD. And so that was not surprising based on the TAN symptoms. Neuropsych testing results also showed significant changes as well. So for executive function, TSC and FTD were both impaired and more impaired than the controls. Language function also showed deficits in both the TSC and FTD groups, and more so than in the control groups. The memory was surprising. TSC patients actually had more memory problems than FTD, and that's actually important because FTD patients typically don't. We couldn't compare this group to the controls because they actually underwent a different test. And then the last domain was visual spatial function, and there was no significant changes between all 3 cohorts. And then going now to the more objective measures of TSC, we did both fluid and imaging biomarkers for TSC and FTD groups. For the biomarker analysis, the imaging showed, we used actually the MRI and the PET scans, which I'll describe a little more. For the cerebral spinal fluid, we really did 4 markers. So amyloid A beta 42, total tau, p-tau 181, I'm sorry, 5 markers. And then the last one, there's 4. So neurofilament light. So for the PET results, we actually used something called Ptowerrelated, FTP, a molecule also known as AV1451. A select few TSC participants completed it, specifically 3. And what we saw was that there was focal uptake in all 3 TSC participants. Specifically in 2 of them, though, the focal uptake of Ptowerrelated AV1451 was seen in structural changes of the brain, which I'll show next. So here in panel A, we're showing the left anterior cingulate as shown by these cross lines here. Panel B is the middle temporal gyrus. The first row is the T1 sequence, the second row is the flare sequence, and the third row is the AV1451. And you can see in the crossbars, more so here in the flare sequence, that there's a hyperintense signal that actually corresponds to the increased SUVR value on the Ptowerrelated scan in all 3 planes. So there is uptake in this, what I would deem as a cortical dysplasia here on FLAIR in the left anterior cingulate. For the left middle temporal gyrus, you also see it's less prominent. It's not as obvious as the left anterior cingulate, but there's actually a focal uptake here as shown in the Ptowerrelated PET scan images. Now, the next one is the CSF results. And so with the TSC and FTD, all 4 markers were done. And you can see that the p-tau was similar in FTD and normal cognition, but significantly elevated in TSC. 32 is actually a pretty high number, and it's actually very similar to the CSF results in people with a MAPT mutation. So MAPT is the leads to the product of tau. And so these people with a MAPT mutation have a p-tau CSF level of 31 picograms per ml. So very similar to TSC patients. NFL levels were also high, but not as high as FTD patients, and definitely higher than people with normal cognition. Total tau and A-beta-42 were actually, there was no significant change in all 3 groups. And this is actually important to point out for the A-beta-42, that TSC does not accumulate amyloid. So it's less likely to be an AD phenotype at this time. Now, to summarize what the findings in the study are, is the TSC symptoms have a broad clinical phenotype, but it primarily involves cognition and behavior. There are psychiatric symptoms as well. There's significant clinical overlap between TSC and FTD. And the problem is that the memory and executive functions are really stereotyped, is what I found in the study. And it really began in the late 40s to early 50s, but it just wasn't well characterized because people haven't recruited these patients for a longitudinal study. So better characterization was definitely necessary. I want to turn now to the current study, which is a neuropathologic study, because there's several aspects of my work in fellowship that just didn't really sit right with me. So from the clinical aspects, the memory was very much more pronounced in TSC patients than FTD patients. And during my recruitment in fellowship, there was no other case with a TSC patient who fulfilled FTD criteria. So over a 2-year span, I was not able to see anyone else that fulfilled that criteria. From a biomarker standpoint, the CSF p-tau levels were quite elevated, more elevated than FTD, but the NFL levels weren't as high as FTD. So kind of a mixed result there, not really sure why it wasn't a slam dunk for FTD. And then the PET imaging, there was that punctate accumulation of AV1451, but unfortunately AV1451 is well known to have non-specific binding, so we're not sure whether that was p-tau or not. And so because of these findings, I, along with Jerry Wang, who's a neuropathologist here, spearheaded this study called Kappa. So it's a co-localization of AV1451 and phosphorylated tau in adult brain tissue. What we're really looking at is, at the cellular level, what AV1451 is binding to in TSC patients, and if it is actually p-tau, further characterizing it with various antibodies, which I'll describe in a little bit. The study cohort and demographics include the TSC group, with an age range of 42 to 58. We also have an AD group between 41 and 59 years old. FTLD group with an age range of 51 to 72, and then the normal control age group between 41 and 80. So TSC and TSC group was actually far younger, and that's expected. And we were trying to age and gender match. So in each cohort, there were 10 brain tissue samples in each cohort. And there were 3 specific brain regions that were requested from the NIH Neurobiobank. And I also want to point out that the gender was equal in each cohort as well. Now, the brain tissue areas, we really picked 3 of them based on their symptoms. So they always had executive, behavioral, and memory difficulties. So Brodmann's area 8, I will refer to as BA8, is involved in executive function. And then Brodmann's area 11, BA11, is the orbital frontal cortex, excuse me. And so that is for changes in behavior. And lastly, entorhinal cortex, which is heavily involved in short-term memory and is actually the first area hit in Alzheimer's disease with neurofibrillary tangles. So that's why these 3 areas were chosen. Now, the p-tau-related antibodies we were comparing to AV1451 include the AT8 marker, which is a general p-tau marker, GT38, which is a marker that recognizes both 3R and 4R p-tau, RD4, which recognizes the 4R p-tau, and then RD3, which recognizes the 3R p-tau marker. So I make this distinction because there are specific neuropathologic findings with the type of p-tau and clinical phenotypes. So for example, the 4R p-tau is actually very involved and heavily seen in progressive supranuclear palsy, cortical basal syndrome, and other 4R tauopathies. The 3R tauopathies primarily PIX pathology as well. And then the 3R, 4R combination is the one that's seen in Alzheimer's disease. So we're covering several different other neurodegenerative disorders and comparing those to the AV1451 to determine the overlap. So I'd like to show you the qualitative assessment of all 4 cohorts in the 3 brain areas. We begin with the AT8 staining. And so you see here the four cohorts are shown here, and then the 3 brain regions, ENTs, entorhinal cortex, BA8, BA11. And so with the controls, as expected, the AT8 showed no staining. TSC showed sparse staining primarily in entorhinal and BA8 regions. The AD and the FTLD tau, as expected, show significant staining in all 3 brain regions, and more so in the AD cohort. The inset just shows you specific cells that were accumulating the tangles shown in brown. And then the scale bar right down here is 200 microns. The next antibody was GT38. And so again, with the 3 cohorts and the 3 brain regions, the controls were not included because it did not show any staining. The TSC patient did show more staining and also involved the BA11 regions. The AD and FTLD tau also showed more staining, but AD, of course, showed the most significant staining as expected. We also try to quantitate these findings as well. Right here on the left panel, this is the semi-quantitation of the AT8 score, and then this right panel here is the GT38 score. So it's subdivided into the 3 brain regions as shown here. So with controls as expected, there was nothing in the BA8 and 11, but you can see that there, in the entorhinal cortex, there's a few areas that showed 1 plaque. And so the semi-quantitative score really just is very simple. So 0 means 0 tangles, 1 means 1 tangle, 2 means 2, 3 means 3 to 5, 4 is 6 to 10, and a score of 5 is greater than 10 tangles seen in a 10x field. And so these bars really show you kind of the spread of it. So this bolded bar really is just the median value of the score. The top portion of the box shows you the 75th percentile. The bottom box shows you the 25th percentile. And so there's a little bit of a spread here in the controls in the entorhinal cortex for the AT8 staining. And then in the TSC group, you can really see that there's a progressive increase staining of AT8. And then, of course, the AD group shows that there's significant staining with a score of approximately 5 throughout all brain regions. Oh. So unfortunately, my PowerPoint has just froze on me, so I'm going to have to restart it. Sorry about that.
Andy, sometimes going out of, presentation mode helps it too.
Does it? Okay, thanks. I think it's back now. So let me get back to where I was.
Yep, we see it.
Okay, great. So we saw the AT8 score, the GT38. I don't know why it's white now, but I think this is this is not the right presentation. So there we go. Okay. There we go. And so, yeah, the AT8 and the GT38 have very high scores in the AD group as expected, but the TSC group is showing increased p-tau staining both in the AT8 and in the GT38 groups. We also did this for the RD4. Again, sparse staining in the TSC group and all in the entorhinal cortex in BA8, but no staining in the BA11. AD and FTLD tau also show staining, but more so in AD as expected. There's because of the mixture. And then the last one is the AV1451 fluortaucipir. So TSC is binding to phosphorylated tau, as you can see in all three brain regions. And AD also shows binding of the tangles, but the arrows are actually showing binding to plaques as expected. There's that off-target binding. So, and then lastly, the FTLD tau shows binding to neuronal tissue, neurons as well. And the scale bar is 20 microns. When we semi-quantitate it for the RD4 and the AV1451, there's really almost no staining both in the controlled TSC until you get to the entorhinal cortex. You see a few areas that are staining positively for RD4, and then there's a significant staining in AD. But for the AV1451, you see actually pretty much in all three cohorts that there's significant staining, and it's more spread out in TSC when you go from BA8 to from 11 to 8 to the entorhinal cortex. So there is a lot of overlap, and it looks similar to the staining of GT38. So in conclusion, what this means is that TAND is potentially due to the accumulation of p-tau, and the subtype of p-tau from the study implicates that is actually the same p-tau seen in Alzheimer's disease, which is a little puzzling. So it's not surprising to me though, because there's just a lot of overlap between phenotypic overlap between TSC and FTD, but neuropathologically, it seems like there's more overlap between TSC and AD pathology. And I think what we have to also note is that there's this accumulation is independent of amyloid. So this implicates, could dysfunction of the mTOR pathway lead to the accumulation of AD-related p-tau? So is TSC just showing us the pathway that p-tau is formed and is actually accumulating? And could this be a potential therapy for AD patients? There is a class of drugs called mTOR inhibitors that are given to TSC patients. And so if given to AD patients, could this actually stop the accumulation of p-tau in Alzheimer's disease? And there's a strong need for this right now, simply because there's amyloid is the hallmark protein, but it doesn't correlate with a lot of clinical symptoms. So 30% of 80-year-olds who have normal cognition actually have amyloid in the brain, but they have no symptoms, but they have AD pathology. The people who have significant p-tau on PET scan or CSF are the ones that track well with decline in their cognitive or behavioral symptoms. And so this is kind of where Jerry and I are really going to describe our findings and hopefully start an mTOR inhibitor clinical trial for AD patients to determine would it help with p-tau in these patients? So this is my references. I'd like to acknowledge everyone at UCSF and here at Duke as well. And thank you for the time, and I'm happy to take any questions.
Thank you, Andy. That was great. I think Wayne's comment is my favorite comment of Grand Rounds during the COVID. He said, that seems to be a perfect K-23 project. Very interesting, although I know nothing about it. I think
Thanks, Rich. Thanks, Rich. I actually do have a question.
Okay, fire away.
Andy, it's a very interesting talk. And so one of the things this is really the first time really kind of learned quite a bit in the last hour. So it's very interesting. You see the phenotype of similarity, also clinical phenotype, and you try to correlate the genetic component and some of the pathology component. But one question I would say, the TSC, one signature of and feature of the disease is really seizure, which would probably cause a lot of kind of clinical phenotype in terms of their behavior components. So how you, and do you think that was one of the cause?
For the elevated p-tau?
Not elevated p-tau is, I think maybe the seizure was really caused a lot of
it's absolutely possible. But I think these are the people with TSC who have very mild disease. So some of them actually don't even have a seizure history, or maybe only had seizures in childhood, and then well controlled on 1 or 2 AEDs, and they were able to live a pretty normal life. Some had JD degrees, some had master's degrees. They were very high functioning, but then they started having the cognitive problems later on in life. So
I see.
Kevin Van Landingham, you had a comment or question?
An epiphenomenon, as we do not fully understand the pathophysiology of AD. No, so I think that's absolutely right. So I think the AD amyloid hypothesis is still the hypothesis, but I think there's a lot of evidence to really describe it. With the pathophysiology of FTD, though, it is pretty well established that there's a lot of FTLD tau, and that's why I really honed in on that group. That group significantly overlaps with AD and with TSC too. So I think the longitudinal study is very important here, which can really help tease out that question.
Andy, Jim asks, what's the average age of the TSC? But I think I could add something onto that, which is you're kind of implying that maybe this has a disease effect, but maybe what it really has is a accelerated aging effect with just these changes because somebody's brain is phenotypically older, not specifically diseased.
Yeah, so that's what you're saying is kind of the point of the study, is that it's just accelerated aging.
Right, rather than a deviation towards disease.
Yeah.
As you know, people in the hippocampus and medial temporal lobe, as they age, have a lot of neurofibrillary changes.
Right, absolutely. Yeah, so absolutely. So I think that's definitely a possibility, and just recruiting patients who are in their 20s and 30s would be helpful to better characterize them from a biomarker standpoint to answer that question. Absolutely. But for Jim's question, I think the average age of diagnosis is actually quite young now. So the epileptologists in the crowd, I think they're more used to seeing TSC patients, especially the pediatric neurologists. So they're typically before the age of 2 because they have such significant seizures and stereotype seizures seen on the EEG. So,
Mike,
when does the?
Oh, sorry, Jim. Go ahead.
When does the psychiatric symptoms develop? If you're seeing the clinical phenotype at age 2,
yeah,
do you start to see them at an early age?
I do. So when I saw them during fellowship, a lot of the psychiatric symptoms, primarily mood, was beginning in the late teens to early 20s. So a lot of them had significant anxiety. A lot of them had depression around that time point. And then they began seeing a mental health provider stabilize, but then later on, several decades later, they started having the cognitive problems. So it was very stereotyped, actually. So,
Mike Lutz.
Yeah, Andy, a great talk. Just a quick question, because I noticed the ages of your AD patients in the study were started about age 41. Did some of these patients have presenilin or APP mutations? Like
yeah, great question. So some of the patients actually were Down syndrome patients. So they did have an extra copy of APP. So,
okay,
absolutely, yeah.
All right, I think the comments and questions finish off with Bonnie. I thought I saw a hand raised. Was that true? Scrolling through.
Rich, this is Jerry.
Oh yeah, hi Jerry, sorry.
So, so just a comment about epilepsy. So just looking at the brain tissue we had, I didn't see histologic evidence of like an epilepsy, like the Shazin gliosis and those epileptic changes. So I think we selected these brain tissues sort of at random, and I don't see like an obvious sign that these brain areas have been affected by epilepsy.
Okay.
Thank you, Jerry.
Okay, so then Bonnie ends it with a couple of clapping hands, and I think that's a good way to end it. So thank you very much, Andy, and the Memory Division for putting on a great grand rounds, and everybody stay safe.
Thank you.
Bye-bye.